So let's talk a bit about the other species of staph, the staph epidermidis and staph saprophyticus. Now they are called coagulase negative staph species because they do not convert fibrinogen to fibrin, or they do not have the enzyme that converts fibrinogen to fibrin, and this is one of the things that separates or distinguishes staph aureus from epidermidis and saprophyticus. And though they are not the only coagulase negative staph species, they are the most clinically important and most encountered. So, quick note that staph aureus and staph epidermidis cause what we call nosocomial infections, and these are actually infections that you occur, excuse me, from the hospital. So generally, staph epidermidis or staph albus, epidermidis is also called staph albus. So staph albus and Staphylococcus saprophyticus are catalase positive. That's what makes them staph. But then they are coagulase negative. That's what differentiates them from staph aureus. What did I eat? And they are urease positive, which means they have the enzyme urease. And when you break this word into two, you have urea and an ACE, which is an enzyme that breaks something down. Now, bacteria actually are able to cause the infections more, or they're able to survive better in alkaline environments, right? And so what these stuff do is that they're able to break down urea into ammonia and water, and then ammonia actually increases the pH of the site of infection to create a favorable environment for them to colonize the sites they are causing the infection. So staph aureus is not urease positive, it's urease negative, and then epidermidis and saprophyticus are urease positive. That's they are able to convert urea into ammonia to increase the pH of the site where they are causing the infection, to give them a favorable environment to colonize. So both Staphylococcus epidermidis and Saprophyticus grow on mannitol salt agar, but they do not ferment mannitol. Like we said, if a bacteria is able to grow on mannitol salt agar, then it means it's staph, or let's say it's mannitol salt agar promotes the growth of staph species. Sorry. But then we can use it also to differentiate between staph aureus and the other staph species, because staph aureus is able to ferment mannitol and then produce a orange color, where the original color or the actual color of mannitol salt agar is pink. Now, you might be wondering, how do we differentiate between staph epidermidis and staph saprophyticus then? Now, you do your test first. You have a gram-positive and bacteria. It's catalase positive. That means it's staph. But then you do your coagulase test and it's negative, which means it's either epidermidis or saprophyticus. Now, how do you differentiate between the two? Now, we can differentiate between the two using an antibiotic we call novobiocin, and um, and novobiocin is able to kill Staphylococcus epidermidis. So we say epidermidis is novobiocin sensitive, and then Staph aureus, Staph saprophyticus is able to resist the killing effect of novobiocin. So we say it's resistant. So when you have a staph which is catalase positive, coagulase negative, urease positive, and you want to differentiate or know the actual stuff it is, we use novobiocin. And when it's sensitive, it's epidermidis, and if it is resistant, then it is saprophyticus. So staph epidermidis causes neonatal sepsis, high yield. Most of the time causes neonatal sepsis, endocarditis, especially when there is prosthetic implants, probably in the heart. What this stuff does is it's able to produce some biofilms which actually coat the bacteria and then prevent the immune system from getting to it and killing it. So it's able to cause endocarditis, prosthetic joints, vascular grafts, and boils. And staph saprophyticus actually causes urinary tract infections, especially in young women. And statistically, staph saprophyticus is second to E. coli in causing, urinary tract infections. So there's a high frequency there. Let's talk about culture, since we are medical lab scientists. Now, we can grow or culture staph species on blood agar and mannitol salt agar, any of them. The conditions needed is, the body's optimal temperature is 37 degrees Celsius, so we grow it in 37 degrees Celsius. But frankly speaking, they are able to form pigments best at room temperature, that is 20 to 25 degrees Celsius, especially the staph aureus, to get like a better golden colonies. I don't know what you do with that, but while they are facultative anaerobes, that means they can grow with or without the presence of oxygen, and so and that is the kind of environment we provide for them. Color of colonies, we have said time and time again that staph aureus produce good golden colonies, and because of the staphyloxanthin it produces, and then the other staph species produce white kind of gray colonies. And then for hemolysis, we said that staph aureus is able to hemolyze or break down red blood cells, and so staph aureus actually causes complete hemolysis of RBC, which is like making it a beta hemolytic, and then they are seen as clear zones on blood agar. So these are the clear zones you are seeing around the staph species, which is showing you that the red blood cells over there have been hemolyzed, and this is actually used to grow some other type of bacteria. I think it's influenza, Haemophilus influenza, which needs heme to grow. So we actually put stuff alongside it so that it hemolyzes the RBCs, and then they get the heme to grow. The other staph species are non-hemolytic. That is to say that staph epidermidis and staph saprophyticus do not hemolyze RBCs. So that is a cultured staph, most probably staph aureus, because of the clear zones. And then this is how they look under the microscope, like a grapefruit, in clusters like that. Treatment. We use vancomycin for MRSA, that's methicillin resistant staph aureus. That is the bacteria staph aureus, which is able to change the penicillin binding protein, and then cause it to be a bit immune to antibiotics. So vancomycin, though expensive, is what we use for MRSA. And then beta lactam drugs such as penicillin and cephalosporins are usually used to treat staph infections, but then they're usually given with beta lactamase inhibitors. Now, staph especially has what we call beta lactamase, a lactamase. That means it breaks down lactam. It breaks down beta lactam, right? And there's beta lactam in the drugs, penicillin, cephalosporins, beta lactam drugs. So it means when beta lactam drugs are taken, they are not really effective because the bacteria is able to break down the beta lactam ring in it, and then renders it a bit useless. So usually beta lactam drugs are given with beta lactamase inhibitors to inhibit the enzyme that is going to break them down and cause them to um, uh, kill the bacteria. So let's take a quick summary. Talking about bacteria, we can have gram positive or gram negative, and for gram positive they appear blue or purple under the microscope uh, because they're able to retain crystal violet after gram staining, right? So under the microscope they can be seen as spherical or rod-like. Now if they are spherical, then they are cocci. It's actually coccus and then the plural is cocci. And then if they are cylindrical or like rods, they are bacilla. That's another one we'll talk about later. And then so gram positive, spherical cocci, rods, bacilli. Now with the cocci, um, whether it's catalase positive or negative, we divide them into staphylococcus and streptococcus. When it's catalase positive, that is containing the catalase enzyme, then staphylococcus. But then if it's negative for catalase test, streptococcus. Staph is usually seen like clusters, clusters like grapes, and streps is more like in chains. Um, Now, when you have staphylococcus, how do you differentiate? You do the coagulase test. If it's coagulase positive, we have staph aureus. If it's coagulase negative, then it can either be staphylococcus epidermidis or saprophyticus. Now, we use novobiocin to differentiate between them. So, novobiocin sensitive, epidermidis resistant, saprophyticus. Thank you and see you another time. Bye bye.